Hello, everyone. Thank you for joining us in another session of the ISCB Student Council webinars. This week, Aarthi will give a talk about understanding metabolic interactions in microbial communities. So please start.
Thank you, Catalina. So let's share my screen. Yeah, I hope you can see my screen.
Yes.
Yes, okay, so let me get started. So thank you once again, ISCB Student Council, for having me in this webinar session. So I am from Genome Institute of Singapore, and I'm currently doing my postdoctoral research. So today I'll be talking to you more about the work that I did during my PhD and how I extended it also during my postdoc. So this talk is going to be about an exciting area of microbial communities because there has been a lot of interest in characterizing and identifying the organisms in a microbial community. So what we are looking at is to understand the metabolic interactions in a microbial community, that is how two organisms in a community interact in terms of the metabolites that they exchange. So, as we all know, microorganisms are found everywhere. That is, they are found in any practical location that you can think of, such as, say, skin, gut, soil, etc. And just give me a second. Yeah. And these organisms are present generally as a community. For instance, if you look at the soil, there are hundreds and thousands of organisms in the soil which act together and are involved in a lot of functions such as regulation of biogeochemical cycles and decomposition of organic matter and so on. Similarly, in our gut, it has been reported that there are a trillion of organisms which perform several functions, including the degradation of complex polysaccharides into short chain fatty acids that can be absorbed by the human cells. So within these communities, there can be a lot of interactions that is happening between these organisms. And these interactions typically help in defining the community structure. And because of the amount or the number of organisms that are present in a community, they often have multiple advantages if you were to use a community in an industrial application. And these are because they exhibit division of labor and also have enriched capabilities. So, because there are multiple organisms present in a community with their own specific function, there can be a division of labor that, say, you have a community of two organisms. One organism takes part in a particular conversion, whereas the other organism takes part in another, in which case there is a complete division of labor. And because of multiple organisms pooled together in a particular location, there is an enrichment in the capability. And this particular microbial consortia, I mean, the microbial consortia in general, have been used for the production of industrially important compounds as well. So this includes, for instance, vitamin C, where you have a consortium of organisms with K. vulgare and B. megatherium, and also biofuel production from lignocellulosic or the plant biomass, where you have three different organisms which basically do multiple functions in converting the plant cell waste to biofuel. So, in all these cases, it becomes important to understand how these organisms interact and what can we do to identify these interactions. And predominantly, the organisms are known to communicate with one another through the exchange of metabolites. And in this talk, I'll just be concentrating on understanding the metabolism of a microbial community. And for that, we look at something called metabolic networks. So let me give a brief introduction of what these metabolic networks are. So if you look at the left panel, you can see a bunch of complex reactions. So these are the reactions that happen inside every typical cell or even a bacterium. So these basically help in the conversion of several chemical moieties into energy or other simpler forms. So the networks, metabolic networks, help us in giving a detailed overview of the metabolism that happens inside the cell. And these networks basically consist of reactions, metabolites, and the association GPRs, which take part in the particular reactions. And the construction, I mean, there have been a lot of metabolic networks that have been constructed so far for several organisms, including those of human gut. And there are two types of metabolic networks. The first one is the draft metabolic network, which is directly obtained from the genome sequences. And there are several databases and tools to get these draft reconstructions. So one of the pipeline or the tool is model seed, where you can upload the genome sequence and get the genome scale metabolic model. And path to models is another database that hosts such draft reconstructions. There are other well curated models as well, which require more of a human effort by bringing in the literature evidences and so on to make a well curated model. And there are many methods to analyze these metabolic networks. And these methods predominantly are classified into two major types. The first one include constraint-based modeling, and the second one is network-based modeling. So the constraint-based modeling technique is a very popular one, where they use something called flux balance analysis. So they basically take the metabolic network and apply a bunch of constraints and find a solution space, and then optimize using linear programming to find an optimal solution. So this type of modeling will help you understand what are the fluxes of the reactions or the pathways that are inside a particular cell. However, to do this, you require well-curated models, that is the models draft metabolic networks, which have been extensively or heavily curated using literature evidences. The second class of methods is the network-based modeling, where you basically take the metabolic networks and try to look at the topology of the network and do something like a simple pathfinding, where you can find out which are the sources of what are the biosynthetic pathways, what can be converted to what, and so on and so forth. So in this talk, I'll be concentrating on network-based modeling, since the tool that we have developed is heavily based on network modeling. And for this network modeling, we take the metabolic network and convert them to something called a graph. So let me go through the little bit of the basic. So the first one, there are different types of different representations of a metabolic network. The first one is a substrate graph, where you have metabolites as the nodes, and based on the reactions in which they participate in, they have an edge. So here is glycolysis, where the initial steps of glycolysis, where the metabolites are connected to one another based on the reactions they participate in. The second type of graph is called a reaction graph, where the reactions are represented as nodes, the square ones, and the reactions are connected to one another based on the metabolites that they share. So here, if you were to look at, again, glycolysis, and R1 is glucose to glucose 6-phosphate. So because glucose and is getting converted to glucose 6-phosphate, R1 has an edge to R2. So this is a reaction graph. So a little more informative graph is a bipartite graph or a hypergraph, which essentially has two different types of nodes, the reaction node and the metabolite node, and nodes of the same type, that is reaction and reaction or metabolite and metabolite, do not connect to one another directly. So if you were to look in here, we have an edge that runs from a circle to a square and a square to a circle, whereas there's no direct edge that links circle to circle or square to square. And in this method, or in this talk, I will be using bipartite graph from the metabolic networks of microbial communities. So now that we have a fair idea of what metabolic networks are, so given that these are the metabolic networks of the organisms, how can we identify the microbial interactions in a community? So when they put together, are there any specific metabolites that get exchanged between the organisms? So that's the first question that we are trying to answer. So for this, we developed a tool or an algorithm called MetQuest, which is essentially a novel dynamic programming based enumeration where you have bunch of reactions which you get, and the pathways are assembled based on the reactions and those which produce the target metabolites given the source. I'll explain it in a little detail in the next few slides. So this algorithm basically consists of two phases. The first is guided breadth first search, and the second is the dynamic programming based assembly of reactions into pathways. So again, I'll be explaining these two in detail in the next few slides. And this tool was developed in Python. It's available as a Python package, and the source codes are also on GitHub. So please feel free to use them and give us your feedback on this. So before I get into the details of what this algorithm is, I would like to highlight what are the features of my algorithm. So it essentially has four very important features. The first one is it requires only the topology of the reaction network, which essentially means that I just need the reactions which are present inside the cell rather than looking at the stoichiometry or the atom mapping and so on, which means this becomes our method is applicable even to draft reconstructions that directly come out of the genome sequence. The second one is that it's simple and scalable to larger metabolic networks, which means that since we are looking at microbial communities where we can have more than one microorganism present in a particular setup, it is very essential that the algorithm is scalable, and we have shown that MetQuest, the algorithm that we developed, is scalable. The third one is it efficiently deconvolutes cyclic and branched pathways. So cyclic, I would like to highlight that the metabolic pathways inside the cell have a lot of cycles. So it is also important that the algorithm does not get stuck in the cycles, and MetQuest can efficiently handle that case as well. And the final one is since the metabolic networks have multiple alternate routes of conversion. For instance, if you were to convert glucose to pyruvate, there can be several routes in which glucose gets converted to pyruvate. So MetQuest helps in examining and identifying all the multiple alternate routes of conversion. So the inputs to my algorithm is a metabolic network that is represented as a directed bipartite graph, which essentially looks like this. And directed bipartite graph is something that I explained in the previous slide. And if we were to look at a microbial community, we basically have two organisms in here, and we connect these two organisms based on a common pool of metabolites through which this common pool, we assume that the organisms communicate with one another. So the first input is the directed bipartite graph. The second is the set of seed metabolites, which is akin to the medium components in any microbiological experiments. And a pathway length cutoff, this is optional, but if you give this, it will just give you all the pathways, biosynthetic pathways of length less than or equal to the cutoff. So let me go over the first step, that's the guided BFS. So we have the metabolic network in here. So that is the input metabolic network. And we start with a set of seed metabolites, and we traverse through this in a breadth first manner. It means that I have multiple layers, like say for instance, in the first pass, I'll go over R1. In the second pass, I will go over R2 and R3. Third pass over R4, R5, R6. Fourth pass over the fourth layer. However, this traditional breadth first search differs in here because we have the notion of two types of nodes, visited node and a stuck node. So visited nodes are the ones which have all the precursors present so that the reaction can proceed, whereas stuck reaction nodes are the ones for which at least one of them is absent. So here, since one of them is absent, this reaction cannot proceed. So we use this particular notation and traverse over this whole graph and finally end up with the scope of metabolites. So scope essentially means that what are the metabolites that can be synthesized given the seed set, that is the initial set of metabolites. And the second output that we get is the set of reactions that can be visited. So the ones that are in blue here. And in the second step, we assemble these reactions into pathways. So for instance, if I were to give the pathway length cutoff of 3, I would end up in a table like this. So I'm not getting into the gory details of the algorithm, but I can tell you that the final output will look like this, where you have a table that runs from 0 till the cutoff that you give, and the rows are the metabolites which are in the scope, and the entries in the cell correspond to the pathways. For instance, if you were to look at M8, 3, you have R1, R3, and R6, which can basically synthesize M8 from M1 and A1. So we applied this algorithm on different scenarios. Here, in the interest of time, I'll just present one microbial community that we analyzed. So this community was previously experimentally demonstrated in another paper, which consists, basically the consortium consists of Clostridium cellulolyticum, D. sulfovibrio vulgaris, and Geobacter sulfuricans. So we took these three organisms, constructed again the community directed bipartite graph, which had roughly 15,000 nodes and 30,000 edges. We first computed the scope, that is the metabolites that can be produced given the set of seed metabolites. And the seed metabolites match the inputs of the experimental conditions which were mentioned in this paper. And we found that around 1,100 metabolites can be produced from the given set of input conditions. We next went to analyze the pathways of size 20 to all the metabolites which were in the scope of, say, cellobiose and other seed metabolites. So cellobiose was the substrate that the paper had used. And we also looked at all the pathways, analyzed them for the metabolic exchanges, and found that acetate, pyruvate, and ethanol were most frequently exchanged, as it was previously shown here. So having this algorithm and a few case studies such as this run on microbial communities, so we then went to, so from the genome scale model, now we have an algorithm that can predict metabolic exchanges. Now we wanted to know if we can identify which organism benefits from the interaction. That is, in a community, is there any particular organism that derives a lot of benefit from the other, or is it just going to be the same? So we wanted to extend the same concepts here, and we moved on and developed another matrix called Metabolic Support Index. So for this, just consider this this panel where you have two individual organisms and the metabolic network shown. So in this case, if I were to run MetQuest on these networks, I would on these individual networks, I would see that in organism 1 there are two reactions which are stuck, and in organism 2 there are many more reactions which are stuck. So we wanted to know if the metabolic exchanges between the organisms can help in relieving these blocked reactions. So if there were metabolic exchanges like this, can there be any reactions that can happen in the presence of second organism which did not happen in the individual network? To quantify this, we came up with this metric, which we defined as the fraction of blocked or stuck reaction relieved by the presence of other organism. So this is the simple formula to calculate it. And in the next slide, I'll just give a small example, the same example here. So considering these two individual networks, we see that the stuck reactions in A is 2, that is the orange squared nodes, and the stuck reaction in B, the orange squared nodes, are 5. Whereas when you put them together in a community, we see that in organism A, due to the metabolic exchanges, all these stuck reactions now become relieved, and there is no more stuck reaction, because of which it becomes 0, whereas B still has some stuck reactions which cannot be relieved by this metabolic exchange. So it's still 3. And when we substitute all these values in the formula and calculate, we see that MSI of A is 1 and B is 0.4, which means that A derives a better benefit from this relationship. So using this, we calculated MSI on several known co-cultures which were previously demonstrated. So here there are, we did an extensive literature search and found several co-cultures. So the first one is a co-culture which is used for producing vitamin C and so on and so forth. So again, we can see that the MSI which is predicted for, say, for instance, the vitamin C consortium. So MSI was higher for K vulgare compared to B megatherium. And the experimental observations also suggest that B. megatherium is a helper strain for K. vulgare, indicating that K. vulgare can or has a higher benefit over, in a consortium with B. megatherium. So we were interested in this particular community because we wanted to carry out few experiments to see if we can validate these predictions. And we also applied MetQuest to see what are the metabolic exchanges that happen between these organisms and which pathways they were involved in. So MetQuest predicted ethanol, acetaldehyde as exchange metabolites amongst others. And these were involved in pathways producing more than 10 amino acids. So to see if we can validate this further in a wet lab experiment, we went ahead and set up a two-stage experiment where in the first stage, we grew one of the organisms. So this is just a schematic where, I grew Saccharomyces cerevisiae on a minimal medium with labeled glucose. And when the glucose depletes completely, we centrifuged and took the supernatant. In the second stage, we had two setups. The first one is an experiment and the second one is the control. And in the experiment, we had the supernatant from stage one along with some YNB and glucose and grew the other organism, that's Pichia stipitis, in this particular setup. And in the control, we replaced supernatant with sterile distilled water and did the same and repeated the same inoculation. And in this, we observed the growth and the GCMS and also performed some GCMS analysis. So what emerged out of our observations? The first one was that the growth curves of Pichia stipitis on control and experiment, when compared on control and experiment, so experiment was quite high, that is nearly 1.38-fold higher compared to the control. And in Saccharomyces, which was grown on the supernatant of Pichia, we couldn't observe any such growth increase. So this clearly indicates that Pichia stipitis benefits from the interaction with Saccharomyces cerevisiae, which was also predicted in our MSI. And to see if the metabolic exchanges, we looked at the average carbon labeling of several amino acids. And here, these are the amino acids that come from ethanol metabolism. And we see that the higher fraction of 13C carbon in the amino acids that arise from the ethanol metabolism. So this essentially indicates that, and this along with the HPLC analysis and other profiling that we did, indicates that ethanol is indeed one of the exchange metabolites that get transferred from Saccharomyces to Pichia stipitis. So given this again, given this MSI and MetQuest, so now from the genome scale metabolic model, we have an algorithm that can predict metabolic exchanges, and we also have some metric that can quantify the benefits. Now we wanted to see if these can be extrapolated to a larger community such as the gut. So in the gut, as I already said, there are several organisms, and we wanted to see how well do organisms in the gut support one another. So for this, we actually took 21 microbial species, which came out of a metagenomic study, where they were shown to be associated with recovery post-antibiotic treatment. And we constructed the joint metabolic networks of these 21 microbial species. That's roughly 210 combinations. And we calculated the MSI values to see how well the organisms can support one another. So what we could very interestingly see was, we constructed an association network based on MSI. So what we could very interestingly see was that there are few organisms that derive a lot of benefit from the other organism. So in this particular network, we have shown the top 10% of the MSI values, and the node sizes in here reflect the number of incoming edges. And there were multiple organisms that help few other organisms in the gut. So we took one of the combinations, that's Bacteroides theta eta micron and Bifidobacterium adolescentis, and we evaluated them further in an in vivo model for assessing the microbiome recovery. And we could see that the BTBA combination, recovery of the gut exhibited a higher diversity compared to the individual ones alone. So these were a few of the applications of MSI that I've just spoken about. And to summarize my talk, so we basically developed a tool called MetQuest that can help in identifying metabolic exchanges and also the biosynthetic pathways that are involved in the conversion of source to the target molecule. And we evaluated MetQuest and ran MetQuest on several microbial communities. The second one was we developed a metric to compute or quantify the support an organism receives from the other. And using this MSI, we could basically show that stipitis derives a higher benefit from Saccharomyces cerevisiae in that particular co-culture and in that given environment. So, and also we demonstrated MSI on another community that's from the gut, and essentially meaning that our framework is quite generic and can be used to understand the microbial interactions and identify the metabolic exchanges between the organisms in the community. So how can this help? Like what exactly can this be used for? So we see this framework that that can be used for designing microbial consortia. Say, for instance, you want to perform an industrial application where you want to use microbial consortia and you want to know which organisms to pick in. You can probably have an informed decision made based on the metabolic exchanges and the benefits that organisms receive. And we can also obtain mechanistic insights like how we showed it in the gut community, that how these organisms can transfer metabolites from one to other and which organism has a higher benefit and so on. And since it is generic, it can be applied to gut, skin, and etc. So as with any methods, there are limitations in here as well. So if you recall my first slide, I told you that it's a network-based method, and it does not and MetQuest does not require stoichiometry or atom mapping and so on, which might become important. And also the predictions which come out of MetQuest is heavily contingent on the quality of the input network. So obviously, higher the quality of the input network, better is the prediction you get. And we don't assign any weights or ranking to the metabolites or the paths. So this can be a future addition that one might want to consider if you want to check which particular pathway is actually active and which particular metabolite is toxic to the cell or non-toxic to the cell or helps the cell and so on. And because of the computational difficulties, it is quite difficult to identify long pathways. But this may not be interesting because of the thermodynamics that is involved in a very long pathway, and cell may just prefer an alternate route to carry forward the conversion. And our method is basically viewing the interactions based on only the metabolism. That's only through the metabolic lens. So there are lots more happening, like there could be quantum sensing, there could be any preferential colonization, and so on and so forth. So those are a few other things that we might want to look at when you want to do a holistic analysis of a microbial community. Nevertheless, our method is useful for getting an idea of the metabolism and the metabolic exchanges that happen between the microbes in a community. So with this, I would just like to acknowledge all the people. So this was basically my PhD work. The first three parts were my PhD work, and the last part was a part of my postdoc. So my PhD advisors, Dr. Karthik Raman, Dr. Smita Srivatsava from IIT Madras, and my exchange boss, who was Professor Lars M. Blanc from RWTH Aachen University, and our collaborator from another department in CS, Dr. Meghna Nasre, the funding agencies, that's DST Inspire for funding my fellowship, MHRD for funding my fellowship, DAD UGC for, again, funding my exchange, and IBSC for funding me throughout. And my current boss, Dr. Niranjan Nagarajan at Genome Institute of Singapore, for having me here and giving me the opportunity to work more on this. So these are these are basically the resources that you might want to look into. So all the things that I presented so far can be found in a greater depth in these papers. So please feel free to take a look at them and just drop me an email if you have any questions. Yeah, thank you, and I'll be happy to take up questions.
Thank you so much for this talk. It was very interesting.
Thank you.
It was really good.
Thank you.
We have some questions.
Yeah.
So the first one is, can one use your method to model the interaction between microbial species and the host, like in human gut, and test which species has the highest MSI for the host?
Okay, so with respect to the host, I believe you can find out the interactions between the organisms and the host, because you do have the human host metabolic network currently. So there is Recon, which has been updated and is available now. So you can take the metabolic network of Recon and the microorganisms in here and see what kind of metabolic exchanges happen. So that's the first part. The second part is the MSI part. So MSI works for the organisms. So you want to see which organisms benefit from the other. So I'm not quite sure what exactly is meant by for the host or something. If there is a clarity on this, yeah, I'll be happy to take it up.
Okay. Another question is, can you comment on the scalability of your methods a little bit further? Like, instead of using 21 species, can you consider hundreds of species?
You can consider 100, yes. But the point is, here again with the 21 species, we did a pairwise analysis. So, and the first results that I present were with a community of 3 organisms. So we did go up to 5 organisms in the community to see which are all the metabolic pathways. But scaling up beyond that is quite difficult. Yes. Yeah.
Do you consider different cellular compartments? Some metabolites might not be possible to exchange.
Okay, so about the compartmentalization, we look at the genome scale metabolic networks, and so you have different organisms and different metabolic networks. So each organism is considered as a compartment, and there are exchanges which come out of the organism that essentially means that a particular metabolite which is synthesized by the organism can be put into the surrounding. So that is the connecting link that we are using. So if you were to remember, I mean, I can share my screen again and show you. Just a moment. Yeah. Can you see my screen?
Yes.
Yeah, okay. So if you were to look at this slide, so in here you have 2 different organisms, and these organisms are compartmentalized. So this organism is one compartment, this organism is another compartment. So only the metabolites that can be thrown out of one organism goes into the common pool, and only the metabolite that can be taken up from this common pool goes into the other organism. So yes, it is compartmentalized. Yeah.
Okay. Sorry, I got lost with the question.
Yeah.
Okay. The last question from Melike is, could microbial co-occurrence networks be a reliable proxy to your MSI method?
Sorry, what networks?
Co-occurrence.
Co-occurrence networks. Okay. So the co-occurrence networks are basically derived from the metagenomic sequence analysis. So you basically tend to say if A is present, B is not present, and so on. Here, we are saying if both of them are present together, which organism derives a higher benefit from both. I mean, so it's a community of organisms, and you want to see which derives a higher benefit. So co-occurrence, I'm not quite sure if it can be used as a proxy, but we could see how it works. We have not tried it so far. Yeah.
Oh, and there is one more question from Melike. Would you expect to see once? Oh, I think it's a part of the question you were answering. Would you expect to see ones that support the community most as the hub species in a Cooccurrence network, or are there more contributors?
Or more contributors to?
That's the end of the question.
Oh, okay, okay.
Oh, well, we have another question.
Sure.
By microbial community, you mean mainly bacteria?
No, actually, microbial community means any organism which is present in the community. So, in fact, when we did our experimental analysis, we used a co-culture of yeasts and not just bacteria. So, it is just the metabolic networks availability that dictates the usability of our algorithm. So, irrespective of whether it's a bacterial network or a yeast network, it's the input that's given. Yeah.
Okay. Should the composition of the community first be determined before studying the interactions, or you're working only on well-controlled cultures?
Can you repeat it again? Should
Yes.
the first part?
Should the composition of the community first be determined before studying the interactions? Do you need to know which organisms are present?
Okay.
Uh,
So
to
to answer that question, we would like the input to be a metabolic network. So if you have the draft genome sequences of the organisms, then you can derive a metabolic network. So in theory, yes, we would like the composition of the community to be known, even for you to get the metabolic networks, leave alone the next stage of doing MetQuest or finding MSI or whatever. So you would want to know the composition of the community to, yes, get the networks and proceed further. Yeah.
but it can be done with microbiome data if you are not sure what is in there, because the second part of the question
Okay.
if it's necessary, if you only are using well-controlled cultures, can it be used in unknown communities that you annotate
Yes.
from?
So from the metagenomic sequences, there are tools that can help you do a taxonomic profiling. So once you have that, I think if you have a good metabolic model or a draft model itself, yes, you can use it. But this would be the series of steps that I follow for using this algorithm from a metagenomic sequencing data where you don't know the composition a priori. Yeah. So it's just one more step that you need to add.
And one last question.
Sure.
The metabolic models considered in each organism are based on different media. How did you generalize them?
Okay, so the metabolic networks consist of a bunch of reactions that happen inside a cell or a bacteria. And for the medium components or different medium composition, that's again an input that we give based on the experimental conditions. So that comes in as a seed metabolite set, which is given to the algorithm as an input. So the metabolic network as such is a generic one. The way you process it, the way you want it to work, is something that differentiates different medium conditions. Yeah.
Okay. Thank you very much
Oh, thanks.
again for this talk.
Thank you.
Um,
Thank you for the
and
opportunity. Yeah.
thank you for participating. And I want to remind everyone that they can follow us on Twitter and learn about the next seminars and how to participate if you want to. Thank you very much.
Thank you. Have a good day ahead.
Bye.
Bye bye. Take care.